In this next part of the lecture, we'll talk about calcium homeostasis. So the overall goal of this part of the lecture is to understand the mechanism of the cycle and understand what maintains calcium homeostasis. And just showing this chart again, this looks at how the decrease in serum calcium will trigger bone resorption, which decreases the bone mass, which increases the serum calcium, which triggers bone synthesis and an increase in bone mass. And how is this regulated hormonally, and what processes are involved in the synthesis and the resorption of bone? So the process of the mineralization or laying down bone is created by vesicles that contain a saturated solution of hydroxyapatite, and this initiates crystallization and the vesicles rupture, and the osteoblasts excrete the proteins, the collagen in particular, as a template to have bone formation. Pyrophosphates can block mineralization, and alkaline phosphatase degrades pyrophosphates. And we start talking about bisphosphonates, we'll see how this pyrophosphate has the bisphosphates are essentially a stand-in, and it prevents this process. So bone is a dynamic structure. It is constantly building and resorbing, and in an adult, roughly about somewhere 5 to 10% of the bone is remodeled over the course of a year. For a child, it can be up to 100% of the bone, but it's definitely in the 80 to 90% percentage of the bone gets restructured in a year. And so this process is constantly happening at different rates in people. And many factors will influence this balance. So serum calcium is one of the biggest regulators, along with the hormones and cytokines will also regulate the remodeling cycle. So this is just a cartoon showing the remodeling cycle. So in the first part, you have an osteoclast, which will resorb the bone, and then the osteoblasts will come along and lay down the matrix to start reforming the bone, and then start mineralizing the matrix, and then the bone will go into the resting phase once it's finally completely remodeled, and then the activation will start the entire process all over again. Keeping in mind that as the osteoclasts resorb the bone, it generates calcium in the blood, and that as you form the bone, you're actually taking calcium out of the serum. So factors that regulate remodeling, things that promote resorption, are the parathyroid hormone or the parathyroid hormone related protein, things like prostaglandin, IL-1, IL-6, and TNF, which keep in mind these are pro-inflammatory cytokines. So anything that promotes inflammation near the bone will also promote absorption. Things that inhibit resorption are calcium, estrogen, calcitonin, TGF-beta, and IL-17. So the primary cells that are involved in remodeling are osteoblasts and osteoclasts. Osteoblasts will build the bone, and osteoclasts will resorb the bone. And one thing that's important to note about that is that these come from completely different lineages. Osteoblasts are mesenchymal cells, so they're derived from mesenchymal stem cells, and the form osteoblasts and then eventually osteocytes, which maintain the bone. But osteoclasts are actually derived from hematopoietic stem cells and really follow an immune cell lineage. So rather than being simply kind of opposite cells in the same lineage, they actually come from different lineages. So this is a very simple picture of how osteoclasts get activated. This entire bone remodeling cycle is incredibly complex, and there are literally pages and pages in the literature devoted to how this works. We're going to focus on some very straightforward and some of the most prominent regulators of this. But this will be, just keep in mind, this is a fairly simplistic picture. So osteoclasts express rank on the surface of the cell, and when the rank ligand or rank L activates or binds to the rank, it'll activate the osteoclast. Osteoprogenerin is a hormone that'll block the binding of rank L to rank. And estrogen is a precursor to the OPG, or the osteoprogenerin. So basically, during menopause, when estrogen is lower, it reduces the amount of OPG that is produced, and so it actually inhibits the negative regulation of osteoclasts, and that's one of the reasons that why there's increased bone resorption in postmenopausal women. So they know from animal studies that OPG and RANKL are critical to regulating the remodeling cycle. When OPG is taken away, and this is generate osteoporosis. It's an osteoporosis model in mice. That's generated. When you take away rank L, you end up with both bone thickening. And keep in mind that rank L binds to rank, and that initiates the resorption of the bone. So when rank L binds to rank in the osteoclast, you end up generating lysosomal enzymes, collagenases, and an acidic pH environment which degrades the bone. So all of these work together to resorb the bone and release the calcium into the serum. So the byproducts are calcium. There's collagen peptides or fragments that are produced from this. Pyrrolidinone cross-link fragments are also produced, and you can detect many of these byproducts in serum or in urine. So that all of these things can be easily detected to show that there is either a normal or an excessive amount of bone resorption occurring. By contrast, osteoblasts are what lay down the bone structure. And so there's a lot of things that will activate osteoblasts, such as cyclic AMP, vitamin D, TGF-beta, IGF-beta, and PDGF. And these will stimulate the osteoblasts along with other factors to induce the remodeling or the reformation of bone. So osteoblasts, when they're activated, will produce collagen. They'll produce alkaline phosphatase, osteocalcin, fibronectin, and many other factors that'll result in the production of bone. So one of the important parts of this, and to kind of reiterate that this is all a cycle of formation and resorption, is that there's a link between bone formation and resorption. I deliberately put this rank L on the surface of the osteoblast because it is expressed in the osteoblast, and when they're activated, the rank L can actually activate the rank, which stimulates the osteoclast to resorb the bone. So in that way, there's a maintenance cycle. And again, this is an incredibly complicated picture. This is just a very basic idea of how this happens, but the rank L is exposed by the osteoblast, which induce the osteoclast to resorb bone. So in this way, if there's no disease state or no health problems, this cycle keeps on basically looping back on itself in a feedback mechanism to maintain calcium homeostasis and to maintain bone health. So bone acts as a calcium store, and there is a significant hormonal control of the calcium homeostasis, in which vitamin D also plays an important role. So parathyroid hormone is one of the most important regulators of this. It regulates the homeostasis of calcium serum, and it regulates the resorption of bone in the body to produce serum calcium. And vitamin D is also involved in this pathway to decrease bone mass and to increase serum calcium. So the distribution of calcium in the body is most of the calcium is contained in bone or in mineralized tissue, and the rest of it is circulated in bound or ionic form in the serum. So the total serum calcium is shown here. It's roughly between 2.2 and 2.6 millimoles per liter, which basically means if you do the calculation for roughly how much blood is in the human body, that is a range of 80 micrograms in the mass of the human body. So basically the feedback mechanism is maintaining the total serum calcium within a range of roughly 80 milligrams in a back of the envelope calculation. Within the serum, about 50% of the calcium is ionized, about 40% is bound to albumin, and another 10% is bound or chelated by citrate or phosphate structures in the blood. And this is just the pie chart showing this, that what is ionized free and biologically active, and then the calcium that is bound to the albumin is non-biologically active. And this is just showing that you have the total calcium and then the rough ranges for the ionized, the protein bound, and the complexed calcium. So when you're measuring serum calcium levels, most of the tests that are performed measure the total calcium. What's really important is the ionic calcium that is physiologically active. So ionic levels are not affected by the albumin-bound calcium, and the decreases in albumin can result from liver disease or malnutrition. But basically, if you want to know what the total calcium is, you have to adjust to the fact that there may be different levels of calcium. So when you look at an adjusted calcium, this really is looking at the physiologically active calcium, and so it's the measured calcium and this correction factor for the amount of albumin that's present in the blood. So Parathyroid hormone is a primary regulator of calcium levels and also of bone resorption. It is an 84 amino acid protein. It's synthesized by the parathyroid gland, and it triggers an increase in serum calcium, and its production is stimulated by low plasma calcium. So basically, as the blood calcium gets lower, it stimulates the production of the parathyroid hormone. And the parathyroid hormone is excreted as an 84 amino acid. It has a relatively short lifetime, and when it's split into an inactive part and an active part of the parathyroid hormone, which has actions in the blood to increase the serum calcium, to decrease serum phosphate, in the kidneys to basically resorb calcium back into the blood, and then in the bone to increase osteoclast activity to promote the resorption of bone to increase the serum calcium. So the calcium sensing receptors are found on many cell types, including the parathyroid hormone gland, as we talked about, to produce the parathyroid hormone. It's also present in thyroid to produce calcitonin, which is a negative regulator of parathyroid hormone, as we'll see, and in the kidney tubules to regulate calcium excretion. So calcitonin, as I mentioned, inhibits bone resorption. It is an amino acid produced in the thyroid gland, and other hormones such as thyroid hormone, estrogen or testosterone, and IGFs will also regulate the calcium level in blood and regulate bone resorption. And this is just, again, the feedback mechanism is incredibly complex, but this is just a very basic way to show how this process works. So if we start on the left-hand side and you decrease the plasma in calcium, it stimulates the production of the parathyroid hormone that increases calcium resorption in the bone. It increases calcium resorption from the kidneys to increase serum calcium, and in the intestines it increases calcium absorption as well. This result will increase the amount of calcium in the plasma. Once it reaches a certain level, there will be a negative feedback, and then calcitonin will be produced, and it'll essentially act as a block on the parathyroid hormone and stop all this activity and actually stimulate the formation of bone. So basically, this is the way in which this gets regulated. It's a slightly more complex picture of the feedback mechanism that we saw before, but this just includes some of the hormonal activity and shows how this is specifically regulated. And again, recognize that this is actually a very, very complex process. So calcium is absorbed in the small intestine, and the active transport is regulated by vitamin D, which is an important role that vitamin D plays. And the transport is based on the relative serum and gut levels. So part of this feedback mechanism is basically regulating vitamin D and regulating how the calcium is absorbed through the small intestine. So calcium is excreted in the urine and the feces, and as we showed that the parathyroid hormone and the calcitonin will regulate the amount of calcium that is excreted and absorbed by the parathyroid hormone, and the large intestine levels are governed by the small intestine. So basically, between the calcitonin, the parathyroid hormone, and the vitamin D, those are some of the primary regulators of serum calcium. So vitamin D plays an important role, as we talked about. It increases serum calcium, it increases the gut absorption of calcium, and it also increases bone absorption. And this is just the mechanism by which vitamin D is made. So, and the important component here is to see that this precursor, the 7-D-hydroxycholesterol, is changed by UV radiation into vitamin D3, and this happens in the skin. So this is one of the reasons why people that aren't exposed to sunlight will end up with a vitamin D deficiency, and this can cause bone malformation and also problems with calcium metabolism. This can also come about through other sources as well, but the exposure through the skin of UV radiation creating the vitamin D precursor is one of the main ways in which this is done. Then there are some regulators which take this into a vitamin D precursor in the liver, and then in the kidney there is the P45-1 alpha hydroxylase, which will finally convert the precursor into the active form, and that is a regulator that takes place in the kidney. So as I said, it is the precursor synthesized in the skin. It's stored in the liver, and then it's converted to the active form in the kidney with the 1-alpha hydroxylase as a point of regulation. So things that stimulate the 1-alpha hydroxylase are parathyroid hormone, low calcium, low vitamin D, and calcitonin. So again, this is one of the ways to regulate and maintain the presence of vitamin D in the body, and to stimulate and to maintain the levels of calcium in the body as well. So it's inhibited by low parathyroid hormone, high serum calcium, and high vitamin D. So again, this is all part of the feedback loop that maintains the serum calcium and the bone health in the body. So really achieving homeostasis is the goal. So what we've talked about right now is how we maintain the homeostasis of the serum calcium, and also the maintaining the proper levels of bone as well. But what's also important to understand is how an insult or a disease state can affect this balance and cause significant clinical consequences, and how that'll affect the regulation of both the serum levels and the bone health. So the take-home message for this is that the driving force, is to maintain a stable serum calcium, and that's really what we're dealing with here. Calcium is important for many things, such as clotting, muscle contraction, and cardiovascular function, and the bone acts as the primary store. And these calcium levels are maintained by various regulatory hormones that we've discussed, and then this is really the why it's important to maintain an acceptable calcium level, and also why it's important in regulating bone health as well.